Good morning and welcome to day 2 of the conference. Please welcome from the local organizing committee, Delphine Chavin.
Good morning, everyone, and welcome back for our second day of SMM 2024. I would like to acknowledge the traditional custodian of the land for which we gather in today, the Aotearoa people. I've had the privilege of calling this place home for the past 17 years, and during that time, I've come to appreciate the deep connection the Noongar people have to this land, sea, and waterways, including the Noble Nara, Noble Yeringan, and Jargara, on which my own research has taken place. I pay my respect to their elders, both past, present, and emerging, and extend the respect to all First Nations peoples here today. Before we begin the plenaries, I'd like to remind everyone to be kind and respectful to one another. We are here to share experiences, exchange knowledge, and learn together in a safe and welcoming environment. If you need assistance, please look for one of our DEI representatives who can be identified by the rainbow ribbons, or visit the registration desk to request support. Additionally, if you would prefer to submit electronically, you can find a link to a form on our conference website or the app. This form can be submitted anonymously. Let's work together to ensure this space remains inclusive and supportive for everyone. Now, I'd like to highlight a few of today's sessions. After the coffee and tea break this morning, we'll return here at 11:30 to congratulate our Wood and Norris Awards recipient. Later, at 5:30 this afternoon, we'll have the first poster sessions in the exhibit and poster hall, featuring presenter with odd numbers program IDs. Finally, join us this evening at 7:30 in the Bellevue Ballroom 1, just after the poster station, for the inaugural of Science on the Shoestring panel discussions with the awardees. Don't forget to vote for your favorite presentations. You can rate them through the app. Also, be sure to check the exhibitor hall and posters, where you can find space to mingle with colleagues during breaks. There, you'll find tasty treats, savory lunch options, and specialty coffees available for purchase. All right, let's start the day. I'd like to now welcome Simon Allens, who will be introducing our first plenary for the day.
Thank you, Delphine. G'day, everyone. Thank you. Bit of respect. It's now my great pleasure to introduce this morning's first plenary session. Richard Connor attributes much of his success to the serendipitous discovery of Shark Bay, Garadagudu, Mulgana Sea country, where he experienced some of the most thrilling moments of his life while observing unexpected dolphin behaviour. Now affiliated with the Florida International University, the title of Richard's plenary is The Shark Bay Dolphin Alliances and the Future of Cetacean Behavioural Ecology. Richard?
Thank you, Simon. And I'd like to thank the organizers for inviting me to speak to you today about the Shark Bay Dolphin Alliances and a bit about the future of behavioral ecology in our field in the face of climate change. Perth, Noongar country, and especially Shark Bay, Mulgana country, are really second homes to me, so I feel enormous gratitude for the time I've spent here. You go straight north 850 kilometers, you find Shark Bay, I think one of the most spectacular places on the planet. Agreed, I'm a bit biased. The Amalgamated name is Gatorguda, as Simon says, which means two waters, because as you see, Shark Bay is bisected by the Peron Peninsula, and we have study sites in both waters. The original study site on the east side of Peron Peninsula right there, out of the Monkey Mia campground, and in the Western Gulf, that's since 1982, and in the Western Gulf since 2007 out of Useless Loop. So I just want to show you what an incredible habitat Shark Bay is. It's World Heritage listed, and it's absolutely stunning place, not just in the water, but on land, spectacular wildlife. Again, you see how privileged we are to live and work in this incredible place. In the beginning, 1982, Rachel Smolker and I hitchhiked up from Perth to what was then a very rustic camp where several dolphins were coming into the shallows to take fish from people, and they still are. And we were thinking, you know, like Jane Goodall habituated chimpanzees to bananas so she could watch wild chimpanzee behavior at close range, we would be able to learn something about wild dolphins in the shallows there. And we certainly did. And I'll talk about that somewhat. But we didn't have a boat that year, but we were able to borrow a little 12-foot dinghy on 2 glassy, calm days and got offshore with our hand-rolled black and white film and photographed many of the characters we continue to follow for the decades to come, including my favorite of all time, Real Notch, here in 1982 on black and white film, and here 2 years before he passed away in 2012. Real Notch, we documented a 30-year career consorting females. I think that's the record. He was a master dolphin politician and a minor god. So here's Rachel and myself out on the boat in the mid-'80s. Before I launch into the Alliance research, I'll just set the stage with a few words about their life history and ecology. They're beautiful little dolphins, only 2 meters long. Here's a pup nursing from its mum. They stay with her about 4 years. But in extreme cases, older mothers will keep their offspring and even nurse them for 7 to 8 years, and it's kind of ridiculous looking. They're about as big as their mothers alongside her in infant position. This is an unfortunate picture. This is the famous female holyfin who came into the beach to take fish for decades. She was very popular. And in 1995, she died after a stingray spine penetrated the left ventricle of her heart. It had been migrating for some time. And this is how we found her on the beach. You see these belly speckles that they get just before puberty that spread in coverage over as they age. So she died at 35. Lucky dolphins can get into their 40s and maybe even a bit older. These guys pursue a wide variety of prey, fish, occasionally squid and crustaceans, throughout the water column, poking around the seagrass and sand at the bottom, catching fish in air, and even up onto the beaches, you'll see. But particular dolphins often specialize more or less in particular foraging habitats and behaviors, and that becomes important. So they also have to worry about bigger fish, tiger sharks especially. Over 70% of our dolphins have shark bite scars. These enormous seagrass beds in Shark Bay support a lot of everything: dolphins, dugongs, sea turtles, and a lot of sharks. So they have to worry about that. Social behavior, we see a lot of it. And just like primates groom each other, the dolphins maintain and form and repair their bonds after a disagreement by petting each other with their flippers. It's very important. Here's a bit of video footage Simon Allen got from his drone of 3 members of the Hooligan Alliance petting each other. We see a lot of this. Right. More intense social behavior often involves body-to-body contact. It can be friendly, playful behavior or aggressive, or the sociosexual category, which runs the gamut between friendly and aggressive. These guys have a classic fission-fusion grouping pattern, which means that individuals in different combinations come together and split up throughout the day, much like we do. This is a pretty big fuse. So it's in that fission-fusion context that we've been studying these multi-level dolphin alliances, male alliances. And I think of it as sort of a window into dolphin social intelligence in the wild. Why? Because it's really easy to see. It's really obvious. It's a kind of behavior that humans can readily see and quantify, including the vocalizations. So first I'm going to describe the 3 different levels of alliances and their functions, talk a little bit about how they form, their ecology, and then studies reveal something about the social cognition of alliance behavior. So I want to acknowledge the key collaborators on the Male Alliance Project in each decade, starting with Rachel Smolker, who started the project with me in 1982. And I can also mention Andrew Richards in this decade. In the 1990s, the big Super Alliance Project with Mike Heithaus and also Lynn Barre, who's in the audience here. And in the 2000s, the biggest project we ever did with Michael Crutzen, and I can also mention Bill Sherwin, Jana Watson-Capps, and Simon Allen, and even Rocky, who helped us out one year there. And for the last 10 years, the team has been Michael Crutzen at the University of Zurich, Stephanie King at Bristol, myself now at Florida International, and Simon is all over the place. Bristol, Zurich, UWA. There you go. And by the way, you can go to our website for photos and pictures and publications and so on. Right, here we are just this past August out doing playback experiments, having a wonderful time. So I'm going to talk about these alliances in the order that we discovered them, level 1, 2, and 3. So first order alliances are pairs and trios of males, and the function of these associations is to form aggressively maintained consortships with individual females that can last from minutes to weeks. In fact, up in Shark Bay right now, there's one that may break the record. I think it's like 60 days or something. So here you see a pair in formation behind the female and a trio behind their female. So we also had 3 males in the 1980s that were coming into the beach at Monkey Maya, and they started herding females in there like there was no tomorrow. And so we were able to observe a lot of his behavior at close range, and we learned a lot about it, including this particular vocalization called pops, which are low-frequency click trains, the peak energy 1 to 2 kilohertz, and they're slow, 6 to 12 clicks per second, that they use as an aggressive threat to keep the female close to them. And if she doesn't approach, escalate aggression. Now this is the male snubby waiting for the fish buckets to arrive, and that's the female Hana he's going to pop at. We should have volume on this, I hope. Volume. Volume. Do we have no volume? Turn it up, please.
She moves, he's in closer.
Oh, we missed it. Anyway, pop, 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 pop. That can do a pretty good job, too. So if they don't approach, sometimes you get escalated threats, and I hope the volume's up on this. So what you're going to see is the female's by, and her calf are by one male, the other male's a bit away. He's not happy about it, so he's going to threaten her with a head jerk and a crack sound. There you go.
Yeah, I'm popping now for 150. And in there. With a head crack. 5150 by bottlenose, and he charges her. And he's probably more intentionally.
So females will sometimes try to escape from the males by bolting rapidly away. That's her clam. They've got clam. It's clam.
Wow.
Who's up front with her?
Wow, bolt, 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 bolt.
Bolt, she bolted. Great. Great. Wow, look at her go.
She's going.
She's going. They're not really chasing, are they?
Yeah, they are.
So because the females will try to escape, the males have to guard her, and they have different ways they do this. And out in deeper water, they often have to spread out to forage individually at the bottom, and we have this incredible triangle formation. They dive away from the female, and they're returning from the foraging dive. They return converging on her. They keep her in the center of this incredible triangle. So in this consortship context, we also discovered a really interesting behavior, an affiliative-friendly behavior that's specific to females. One female swimming slightly behind the other in that staggered formation with her pectoral fin resting along on the side of the other. And in this context, where they are being harassed by obnoxious males, we think it's an expression of support between the females. So second-order alliances. We have learned over the decades that this is the core social unit of male alliance formation. Once they're formed, these can be stable for decades, individuals gradually disappearing over time. Rarely will an individual join or leave, sometimes get kicked out, and those are fascinating cases, but these are really stable groups. Here you see a pair with a trio, and here you see a trio behind it, their female, with a pair behind their female. So I want to show how obvious these associations were in the 1980s. Rachel Smoker discovered the first and second level associations. This is video she took about 1986, I think. Look at that incredible synchrony, right? Two trios, side by side, going synchronously. Sometimes when they're excited around a female, two males will get side by side from different trios and surface synchronously. We think that reduces tension in a sort of competitive situation. Say more about that synchrony later. So we didn't know what these associations were about. And it's a real puzzle, because if males are simply competing for access to estrus females, they shouldn't be hanging around together in a friendly way, but they were. So we consider the biggest, most important day in the history of our project, August 19th, 1987, we discovered the function of second-order alliances. The three males that came into the beach at Monkey Maya were herding the female holyfin. Another trio who never came in there came right into the shallows, approached them within a few meters, and did nothing. They left. So what's going on here? So I got in the boat, found them a kilometer offshore. They joined up with another pair who had a female. One of those males was real notch. And together, the two alliances came back in. They charged right into the beach in front of the tourists, and the fighting just exploded right down the park through the mooring lines. And here's a video clip where they emerged at the end of the park victorious.
Holyfin, they went in there and stole her. Holyfin's in the middle of Toxoplasmosis and real notch high and munch and all these guys. I don't believe this.
So troops invite when got reinforcements, huh?
This is like science fiction.
So the science fiction comment was because I had been steeped in primatology and reading the literature about primate societies. And I knew that multi-level alliances were so important in our own species, right up to the level of nation states. But there were no similar examples in all of primates and other mammals. So I thought, wow, it just blew our minds. So it was very exciting. So we were thinking that maybe there'd be a relationship between the size of the second-order alliances and the stability of their first-order alliances, because some males are in the same trios all the time, or pairs, and some males will switch partners between consortships. But it turns out we didn't find such a relationship. There's just a lot of variability there. But I want to point out that 3 different groups have achieved a size of 14, and each group maintained that size for 3 consecutive years. And how thrilled were we this summer to photograph the male gripe, who was part of the original 14-member super alliance we studied in the 1990s, starting in 1994. Here he is 30 years later, hanging out on the edge of a fight. He's the sole survivor of that group. And I'm sure he was putting his 2 cents into the disagreement there. So this shows you these cluster diagrams, color-coded by second-order alliance, shows you just how variable bonds and relationships are within these groups. Some have quite tight bonds, but there's just enormous variation. In particular, males don't spend a lot of time with each other in these second-order alliances. I want to emphasize, sometimes they're all together, and it's really incredible to see that, but often they're spread out from each other at some distance. We also found that the stability of your first-order alliance predicts how often you're going to be seeing consorting females in these groups. This suggests maybe something like dominance relationships. At any rate, each of these second-order alliances is its own little complex society, contained complex society, nested within this huge fission-fusion society and overlapping with a bunch of other little second-order alliances. Livia Gerber, working out of Michael Crutzen's lab, found that males that had the strongest bonds and the most homogeneous bonds had enjoyed more paternities, the popular males, if you will. So look at the central panel there. Those colored shapes represent the home ranges of particular second-order alliances. And you see that each alliance overlaps with a number of others. And as you'll see shortly, not all of those interactions are hostile. And these male home ranges will overlap with individual female home ranges. This is data collected by Jana Watson-Capps out at Janet Mann's lab. And Jana found tremendous variation in the size of female ranges, an order of magnitude, which we think is related to these individual foraging specializations. But the point here is that individuals down here will never encounter individuals up here. It's a continuous open society. That's very unlike terrestrial mammals, where mammals that have complex social relationships typically live in bounded groups that are familiar to us by names like a chimpanzee community or a wolf pack or a baboon troop. So this kind of open society would really confuse this poor monkey. So just like in the 1980s, we were wondering about these associations between first-order alliances and what was going on. We started seeing friendly associations between second-order alliances, and same question. Well, it turns out that second and third-order alliances have exactly the same function: to attack and defend against attacks from other alliances in contests over individual females. Well, if the two levels of alliances have the same function, why is there another level? We think it's an insurance policy, because again, these guys are often spread out from each other. If you are the female and away from your second-order allies and you're attacked, it may help if you have other friends around. And we have some evidence in support of this. Males that have third-order allies can hold on to their females for longer periods of time. So my grad student, Whitney Friedman, was in the middle of studying the relationships, the third-order alliance relationship between these two, the blue PD group and the green KS group. And incredibly, in the middle of her study, the rascals, the red rascals there, who had been in the area forever, suddenly joined and formed a third-order alliance relationship with those guys. So look at that central pattern panel there, and you see the sort of three big blue and green dots indicating that the strongest associations maintaining these third-order alliance relationships are between a particular trio in each group. So you might think that kind of simplifies things, because it's really, these third-order alliance relationships are really just about the relationships between particular individuals in each group. Except when they get together, everybody is petting everybody. So everybody's playing a role in maintaining these third-order alliance relationships. And anyway, that pattern wasn't so evident in that third panel when the rascals joined. And notice how only 3 of the rascals joined. One of them died, and the other one, Lando, was kicked out. Evidently, he didn't fit into the new set of relationships. Poor Lando. So we see conflict at all 3 levels of alliances, between males in first-order alliances, second-order alliances, and third-order alliances. In 2006, I was watching this big fight between the PD and KS alliances, and I thought, oh, this is terrible. They've been friends for so long, and now it's over. The next day, they were back together again. So here we have the PD group is going to join the KS members, and that guy right there, who's kind of jerkily moving forward, is Big Midgie Bites. I don't know why it's so jerky. But Deet is in the other group, and Deet is petting with his mate because he knows what's coming. Big Midgie Bites has an issue with him. Bam! And they chase Deet away, and we didn't see Deet with them for a while. So alliances are formed when males are in their early to mid-teens, not with relatives, but with similar-aged peers, sometimes a male who's a bit older. But the process starts much earlier during the juvenile period. You go out there, especially in the mating season, you look at juvenile groups, and they have these incredible play groups, which are play consortship behavior. They're in pairs and trios, and somebody is in sort of a female role. It can be a male, can be a female. They switch partners. They even have the pop vocalizations. They do a lot of this. And at the end of the day, they go home, and they're different ways. Whereas in adult consortships, they continue to consort the female through all behavioral activities. So Katie Holmes studied these juvenile groups, and 2 interesting findings to focus on here. The first, she found that older juveniles engage in more of this consortship play behavior than younger ones. And that's unlike play between individual primates, because they do it a lot when they're younger, but as they get older, their dominance relationships become more important, and it's more difficult to have symmetrical play interactions. But the dolphins are engaged in team play, which is going to be important when they're adults, so they do more of it. The other really big discovery here is that the proportion of time playing in the actor role, not being it, predicted paternity success. This was the first demonstration that play behavior predicted reproductive success in any mammal, so we were very excited about this. And we were able to do this by using data collected in the 1990s by Amy Samuels and Cindy Flaherty-Stam. So this is really special. We combined that early data with the late data because we lost Amy in 2008. The ecology of alliance formation, we think, is related to these individual foraging specializations. And I'm just going to show you 4 of the most fabulous ones, starting with this sponge carrying. Stephanie took this incredible picture in August. So sponge carrying is a learned vertically transmitted behavior that only occurs in deep channels, mostly by females. It's been studied, it was discovered by Rachel Smolker, and it's been studied extensively by our group and also Janet Mann's group. And funny that every time anybody publishes a paper on sponging, the media, the press acts like they've never heard of it before. Tool use in dolphins. And tool use because they use these sponges to protect their snouts as they poke along the bottom trying to scare up fish. So at the top of the peninsula on a steep beach, we have this incredible hydroplaning behavior that's also mostly females and passed on to their calves. So I hope you can see the fish here that Simon got this drone. You see the fish right in there in the shallow water, the dolphin's tracking it. Fish probably thinks it's pretty safe in there, right? Boom, got it. And it's just to show you they do beach sometimes. Another wild behavior is they'll come up out of the water with these big shells on their noses. What are they doing here, shaking the shell? Well, photographs revealed there's a fish in there. They're shaking the fish out of the shell. And I just want to draw your attention to a Netflix documentary that's going to be later this month. Has some really wonderful footage of the shelling behavior underwater, and it's narrated by Barack Obama. Don't we miss him? And the last behavior here is kerplunking. This is shallow water seagrass beds.
Good luck.
Listen to this. Listen. That pulse sound we think they use to scare fish.
Straining sound is incredible.
Yeah, and yeah, they sound like they're constipated. All right. So one possibility is that these young males form alliances with males with whom they share favored foraging areas and tactics. And that makes sense because then you can coordinate your alliance behavior more easily. So 2 lines of evidence for that. Orla O'Brien, my student, was looking at alliances with greatly overlapping ranges and where they preferred to forage. And so, for example, these 2 alliances, this is just east of Monkey Maya, in what we call the subdivided habitat. So the Hooks crew liked to forage, and they were kerplunkers over these shallow seagrass banks, the green ones, and the X-fins foraged in the deeper channels. And then out in the western Gulf, Manuela Bezozzero found a sponging alliance. So yeah, it's mostly a female behavior, but sometimes sons of spongers become spongers, and then I guess if there are enough of them, they can form a really formidable, I'm sure, sponging alliance. We also found systematic behavioral changes in alliance behavior going up from that subdivided habitat up the length of the peninsula. So in that subdivided habitat, you get a combination of pairs and trios, and north of that, it's almost all trios, and they also consort females at a higher rate, and they have more fresh tooth rake marks indicating higher levels of competition. Well, we had a lot of different hypotheses to explain this, but Pernille Sorensen just published her work, a meticulous study, showing that the key factor here is the acoustic active space of that pop vocalization. Not whistles, pops. So you're a trio of males, you're out in open water, you want to threaten your female, pop, 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 pop. Well, you're also letting everybody else know that you have a female. Maybe not always a good idea. So if we're going to think about social cognition and alliance formation, what better place to start than that incredible synchrony, right? Synchronous diving, incredibly precise, even synchronous side-by-side leaps. And we also see amazing synchronous displays. We think these are creative. There is such a variety. We think they make them up. The alternative hypothesis is that, they get together and, hey, maybe let's do number 7B. That seems unlikely, so we think these are creative. Look at these two males doing synchronous belly slaps going in opposite direction on either side of the female. Triple leap. Whoa!
Wow, sync leap. How cool is that?
It was very cool. This is a rooster strut, and you will see why it gets this name. And one of the wildest we have ever seen this summer. Simon got this on the video. What you're going to see here, there's two pairs of males behind the female and her calf. They're going to emerge from the bottom of the screen. You're going to just see them just completing what we're calling, obviously, this fish whack display. Then they're going to accelerate towards the female and repeat. Do you like that pose? They also coordinate and synchronize that pop vocalization. So at the top, you're seeing a single male and pop trains by a single male, but sometimes two males will pop at the same time. We think coordinating their guarding behavior. And Bronte Moore found that they will coordinate the inter-pop interval when they do this, even when that inter-pop interval is changing during the course of pop trains. So how are they doing this? We know from captive studies that dolphins are phenomenal at imitation in both the motor and vocal domains. In Lou Herman's lab here, they've told the dolphin to do what the human does, and you see they do a pretty good facsimile. So we would like an explanation that unites all of dolphin synchrony, the side-by-side simple synchrony, and these complex looping displays. One possibility is imitation. But Lou Herman and our group both looked at frame-by-frame analysis of this synchronous behavior and found no evidence of leadership. Another possibility is entrainment. And entrainment is when, like, you start moving to the beat of music, or you fall into step with somebody you're walking with. You entrain to their rhythm. But I think it's hard to use entrainment to explain these displays. Well, when Laura McHugh and I were digging through the literature for this more recent paper on synchrony, we found this incredible paper by Noe et al. They were studying human synchrony and comparing the context where one person is imitating the other or where they were both imitating each other simultaneously, mutual imitation. They called it the mirror game. And human synchrony was much more precise when they were mutual imitation, when they were playing the mirror game. I think the mirror game, mutual imitation, provides potentially a universal explanation for dolphin synchrony, which is the only other case of synchrony as an alliance signaling system in mammals, but outside of humans. So when Stephanie King joined us, she bought this boat-based hydrophone array that could triangulate onto localized sounds. Finally, we can tell who's talking. So here's just a quick bit of a video of her collecting data in this context. Or not. Let's see here. Well, anyway, it's okay. So the first thing that she did was demonstrate that each male had their own individual signature whistle. There'd been some question about that in Shark Bay or whether they had an alliance-specific whistle, but no, each male has their own signature whistle. Once she built up a catalog of these, then you can start doing playback experiments. Simon puts the drone right over the dolphin, play back the whistle through the speaker, and you can see the dolphin's instantaneous reaction. Amazing. And we found that males respond more strongly to their second-order allies than their third-order allies. Okay, that's not surprising, you think, except this response is irrespective of bond strength within second-order alliances or recent consortship history. Remember, bonds vary enormously in these groups, and we think what this tells us is that they treat all members of their alliance, regardless of their bond strength, as important team members, allies. And there's two other lines of evidence indicating that they value all males in their alliance, even those with whom they have weaker bonds. So Emma Cheriskin did this work here showing that dyadic bond strength predicts how often two males pet each other. That's not surprising. What is surprising is that whistle exchanges occur more often between males that have weaker bonds, perhaps in lieu of physical proximity. And then, looking at the synchrony, we found that males with weaker bonds actually have more precise synchrony. They're not together as much, maybe they work at it a little harder to maintain their bonds. So, convergence with humans in three important ways. This synchrony as an alliance signaling system, likely related to convergent abilities to imitate in the vocal and motor domains. Vocal exchanges of individual whistles, which is also important in humans. I don't know if it's anywhere else, in lieu of physical contact. And again, these intergroup alliances. The human model had always been based on human social structure. Humans have an intergroup cooperation, and it's always been thought to relate to their pair bonds and knowing who your in-laws and relatives are in other groups you're going to cooperate with. But the dolphins give us another route from a more promiscuous chimpanzee-like mating system to intergroup cooperation. So here, just admire this largest alliance system outside of humans. There's 120 males there in their color-coded second-order alliances next to third-order allies who have similar colors. Just an incredible system within which we can examine dolphin social intelligence in the wild. So now I want to say a few words about climate change. How am I doing? 3 minutes? Cool, perfect. So the question I asked in my abstract, if we lose a population of cetaceans, what will we know about what we have lost? And in most cases, just that there was a population there. And really no more than that. So if we look at this accelerating climate change, I'll just look at primates for a second, right? The golden age of primatology, learning all about their societies and cultures, was from the 60s, 80s, and so on. So if they're in trouble too, not just from climate change, lots of things like agriculture and so on. But if you lose a group of primates, you know what kind of society you've lost. You know what kinds of cultures you've lost. We are potentially now getting into a golden age of cetology with this incredible new technology. I mean, even in Shark Bay, the discoveries we're making now remind me of that early period of discovery in the 1980s. But more importantly, all these new toys are giving us this incredible access to all these other species and populations we've never been able to learn about before. But we're in a race against time. So I think there needs to be a really massive effort to learn about cetacean societies and cultures. But there's two big roadblocks here. Funding is obviously one of them because, you know, let's face it, the funding for behavioral ecology just isn't what it used to be, right? And also, I would blame the fossilized structure of the academy, the department organizations. There's just not really a natural academic home for us in this area. So if you look at, if you go out in the forest and study monkeys and you publish a few papers, you can apply for anthropology jobs all over the world because they hire primatologists because they're interested in homology and divergence, right? How similar or different our ancestors are from us. But that's ignoring the equally important convergence. And we have the evidence now. Every time you study a new species of cetaceans, you find something really remarkable. Look at the bottlenose dolphins, multi-level alliances, menopause in killer whales, symbolic marking in sperm whales. All of these things are convergences with humans. The goods are there. The evidence is there. So, and then we have all these other features which are rare or unique compared to terrestrial mammals. So we really need to have a massive effort somewhere in the academy, ain't gonna be in the US now, but maybe a Max Planck in Germany or something dedicated to cetacean societies and cultures, or maybe just a friendly billionaire or two, I don't know. But need to do that. So buy my book. And I want to acknowledge all these funding organizations, and special shout outs to the Monkey Maya Dolphin Resort who supported us for decades there, the traditional Malgana owners of the region up there, and to Richard Hulst and Joe Haymans, who are back there, who gave us a home in Perth for 30 years. Thank you. Oh, you're not supposed to see that. How much under was I, Simon? All right.
Thank you, Richard. That was sublime. I just want to clarify when Richard says all these fancy new toys, he means really important research tools. We have a full 5 minutes for questions, so if people want to approach the mics up there near Tillen over here. And
I think I'm supposed to walk up so I can hear better.
Hello? Should be all right.
Yep.
Should be all right.
So I was wondering, what do you think the females think about all this?
I think, and we published on this, I didn't have time to get into it today. We think the female strategy is to mate with as many males as possible to confuse the issue of paternity because of the underlying risk of infanticide. So they probably want to mate with each male, but they don't want to be monopolized by any male. And then within that, getting at female choice, that's difficult. We've tried it before. I think Janet's group tried. It's a very difficult question to get at, and it might be internal. And so that'd be totally hard, but and maybe, yeah.
Any other questions from the audience?
Hi, thank you for that talk. It was awesome. I was just wondering if there's any way to look at sperm competition among the males in the different alliances.
Yeah, that's a great question. We'd love to. You know, to get at the question of, for example, skew, reproductive skew within these trios, you'd really need to have a large sample of each one. You'd need to have one alliance you're watching, hopefully a stable trio or something over decades. But in general, by sampling a lot of males, and if we keep at it, we may be able to relate paternity success to other, you know, like we do with the bonds, for example, and to other various characteristics. But sperm competition, yeah, it's probably there, but I don't know how we would quite get at it.
Good morning. So I have a question. How do you see these systems translated in other dolphin Tursio populations or in other small cetacean species? Do you think they could follow the same systems or the same structure, or is a specific structure to the population in Shark's Bay?
We know there are differences, like they're very different from the larger bottlenose dolphins and the common bottlenose dolphin in Sarasota and so on. And that's why we need to get this big funding to go out and study all these other populations, right, and species, to find out how social structure relates to ecology, body size, et cetera, et cetera. So I would love to see a massive effort along those lines.
Thank you.
Thanks for the great talk. Just a question about the sex ratios in the population. Is it a male-biased adult sex ratio, and how would changing sex ratios affect the alliance?
I think it's about equal, but remember the females have this long interbirth interval, so at any given time, a proportion of the females are available to males, and so that's probably why they form these trios. Hal Whitehead and I made a model which, when you put in a few reasonable assumptions, you get pairs and mostly trios out of it when they're competing for a certain number of females. The competition's a certain level. The new model I was going to talk about but didn't, I think explains the multi-level alliances, and I can talk about that individually with you later at some point.
Hello. Thank you so much. That was a great talk. If you're looking for a postdoc, hello. But my question is, within these alliances, do they take turns with the female to try to make babies, or is there a hierarchy, and who is then chosen for being the leader and able to actually mate with the female?
Yeah, we don't know. And one of the problems is that we see a lot of sexual behavior, but we see tons of just non-conceptive social sex in this population in general. And probably a lot of it occurs further underwater where we can't see, and maybe the drones will give us more insights to that in the future. We'd love to know that too.
Thank you.
Mm-hmm. Great question. Mm-hmm.
Yeah, hey, fantastic stuff. Thank you, and well presented. I think I'd be misinterpreting you if I took this, at least if they go extinct, at least we knew. I don't think that's how you meant it.
Yeah, absolutely, yeah. I mean, if you know, we had this big heat wave in Shark Bay in 2011, it did a lot of damage. Now imagine if we'd never studied up there and there was lots of heat waves and basically the population got reduced to very little and all this complexity was gone. If we hadn't studied them, we'd never know it was there. So yeah, what I'm saying is that at least for posterity, we need to find out what all these different societies and cultures are like out there, because you know, if we're being realistic, we're not going to save them all. And even if there's remnants of them, they're not going to be what they were when they were at their full population size.
So cool. So I guess my question is, what would you have to say about leveraging the greater understanding of them towards actual, you know, action to protect them and less known whale and dolphin populations?
Well, I think we can use these exciting results. We can go out there and say, look, every time we study one of these species in any depth, we just find something amazing, you know, and just use that to just convince people of how important this is, that we need a massive effort in this area, and then you can kind of study everybody. And you know, there's a long history from primates and others where when you get a study is going on a charismatic megavertebrate in a habitat, it tends to sort of automatically have some protection for everything else there, right? So there's some of that, but you know, against climate change, that's a whole other level of problem.
We're just getting over time now, so perhaps we have time for one more quick question. And then, of course, Richard will be around for the week. Yeah, go ahead, Johan.
Hi, thanks for the presentation, especially for the enthusiasm and the engagement that you give in your presentations. I'm very curious about of course, all these alliances, but you said that from time to time, one of the members of a certain group or alliance is sent away. I mean, it's refused by the group. Do you think, I mean, is there any kind of theory that you might have how this mechanism triggers? Suddenly somebody is not cool anymore and is kicked out?
I think it often happens in the context of new relationships forming, because just like that case I showed you with Lando, we had a case in the 1980s. If you read my book, the poor male Lucky wasn't very lucky. He kept getting kicked out of alliances, and often in the context of other dolphins forming newly formed alliances and relationships. So yeah, Lucky wasn't lucky. Although I think the paternity data showed that he did get lucky. I don't know.
Thank you very much. Could you please join me in thanking Richard for that sublime plenary.
All right, thank you. Thank you.
Not to take from Richard's pitch for large sums of money from you, but there's evidence from various other populations of Oduncus, Luciana Muller's work and students in Port Stephens, also in South Australia, and I think there'll be some talks later from Quincy Gibson and her group, St John's River Animals. So there are other alliances out there, not just Shark Bay, even though I'm biased towards Shark Bay. Also across the north of Australia, we see some evidence of alliance formation in Australian humpback dolphins. Thanks very much, Richard. Totally brilliant. Our second plenary of this morning is by my dear friend, Rochelle. And anyone who knows either of us will have no trouble in predicting which particular line of her bio that I'm going to highlight this morning. Dogs are her favourite people. Rochelle comes to us from the University of Auckland, and the title of her plenary is The Māui Dolphin, a cautionary tale of inaction.
Oh my, there are a lot of you. And no, Bex, I didn't get a box so you could see me over the podium here. It's not built for shorties like me. Now, unlike Richard, I'm working on some animals that have a very different story. So let's go. The Māui dolphin. G'day. I acknowledge the Whadjuk people as the traditional custodians of the land on which I stand today to tell the story of the Māui dolphin. It's the world's rarest, most endangered, I would argue, fully marine dolphin. These dolphins are a taonga, they're a treasure, to the tribes of Indigenous peoples in Aotearoa, New Zealand, who are kaitiaki. They're the guardians of the west coast of Te Akea Māui, the North Island, and this is the home for the Māui dolphin. The loss of Māui dolphin is greatly felt throughout the loss of cultural connection to these people. The work I'm going to talk about today is not just mine. We all succeed because we stand on the shoulders of giants and because we live in a very big village. Many of the faces here are the hardworking people, past and current, working for these dolphins. There's a lot of edited highlights in this talk. There's a lot of complexity that I'm not covering. It spans a lot of really difficult things like disagreement and decades of disagreement. And so within that, there's always nuance. But I refer to a lot of the papers, and there's a lot of literature, so go and check it out. I'm hugely grateful that I get to work with these amazing people, and in particular Scott Baker and Debbie Steele, who I've had the privilege of knowing for many years. These are the Māui dolphins on an uncharacteristically glassy, calm day. Oh, you can turn the sound down. We don't need sound. Please enjoy. They have magnificent lives. They have no idea they're nearly extinct. And it's a great joy every time we go out that they're still there. It really is. This is the rugged, mostly inhospitable coast. That's the core habitat for the Māui dolphin. It's not Shark Bay. It is the antithesis of Shark Bay. Once frequently sighted off the south of Taranaki, on the west coast of the North Island, Te Akea Māui, Aotearoa, New Zealand. Their current range is restricted to around 250 kilometres of coast, from Kaipara in the north there, down to Whangaroa or Raglan in the south. It's a fraction of their historical range. Almost all the data we have is from summertime, and it's a really tough place to do research. They are the smallest fully marine dolphin in the world, and they're endemic to Aotearoa, New Zealand. The Māui dolphins separated from their South Island cousins, the Hector's dolphins, around 20,000 years ago, but they look exactly the same. You cannot tell them apart when they are side by side. They socially aggregate near to shore in summer, and they mate and calve and forage. They have quite a fun time. They're right in close to shore. When autumn arrives, they disperse, and they head either alongshore or offshore in smaller numbers. Once upon a time, they would have been an important part of an ecosystem, but today Māui dolphin probably play very little role in a functioning coastal ecosystem. They are the only fully resident coastal dolphins along the west coast of the North Island. The dolphins didn't have the knowledge to avoid monofilament nets that gained popularity in the 1970s for recreational and commercial fishers. They were extensively used along the west coast of the North Island until the early mid-2000s. By fishers. There is patchy information about the historical distribution of Cephalorhynchus. They belong to the Cephalorhynchus hectori group. So they're Cephalorhynchus hectori. Hectori is the South Island, and Cephalorhynchus hectori Māui is the Māui dolphin on the North Island. There have been very, very few samples in which we've been able to use to genetically confirm species ID. We have historical museum specimens, beach cast and or bycatch samples, and one from the stomach of a 7-gill shark that was caught in a set net in the Manukau Harbour. So it's like a double whammy catch, that one. Despite numerous reports of Cephalorhynchus along the east coast, No genetic samples have been taken until recently, when a small biopsy sample was taken from an adult accompanied by a large juvenile right inside the Hauraki Gulf. So it's on the east coast, but right down in an area where they really are not often seen. So we managed to get a genetic sample, and that animal is a Hector's dolphin from a CA. And we think that it's almost certainly from the east coast of the South Island and has come up the east coast of the North Island. This meagre amount of information has been critical to tempering the often fractious debate about the Māui dolphin. We simply have too little data to be able to say what we need to be able to say in meetings or in arguments. There were only ever hundreds of Māui dolphins. Several pre-decline modeled estimates suggest there are around 300 to 500 dolphins prior to the introduction of set netting. In the mid to late 1980s, there was serious concern about the levels of bycatch. A single boat survey conducted by Steve Dawson and Liz Slutin in 1985 estimated around 134 dolphins. The use of genetic tools was introduced by Scott Baker in the mid-1990s, and in 2002, the North Island Hector's dolphin was recognised as being distinct from the South Island Hector's dolphin and renamed the Māui dolphin, after the Māori ancestor Māui, who fished up Te Ika a Māui, the fish of Māui, which is the North Island of New Zealand. As I mentioned, historically, almost all reported beach-cast or bycatch dolphins along the west coast were not sampled, so they could have been either Māui or Hector's dolphins. We will never know. Bycatch caused a decrease in their range, as you can see here, through the decades, shifting the southern extent to north of Taranaki. Of 45 dolphins in the stranding record throughout this time, the cause of death was determined for 14. 7 had marks consistent with entanglement, and another 4 had cuts on their bodies or slit bellies consistent of removal from nets. There were no doubts that fishing was the problem. With the success in the 1990s, extracting DNA from the few historical samples that we had, working out whether it was a Māui or Hector's dolphin, it was clear that genetics was a critical tool in the research toolbox. Back then, mitochondrial DNA was the best thing ever, which may be amusing to those of you in the audience that were born this century, but it was revolutionary at the time. There was a lot of excitement in our lab. Genetics has come a long way, there's no doubt about that. And we are now using genomic approaches in our research. Some people raised concerns about biopsy sampling, and so we trialed scrub pad sampling, but the DNA was frequently very poor quality and had a very high, rate of failure where we couldn't say anything. Unfortunately, there was misinformation, and there was strong opposition by some researchers and NGOs to the collection of biopsy samples and the new genetic approaches, and research was unnecessarily delayed for many years. We lost valuable time, but eventually returned to using biopsy samples, and for the past 15 years, thankfully, it's been a key research method. So far, we have used that tiny sample of skin, and sometimes blubber, for the following projects. And we still have tissue left from most of these samples for future work yet to be imagined. So any of you budding young scientists out there, if you can think of something really cool to do with a teeny tiny piece of Māui dolphin, get in touch with Scott or Debbie, not me, or Emma. I'm genetics adjacent. So Hector's 101. We know that Māui dolphins have only haplotype G, and we've looked really hard to find that haplotype G elsewhere. Despite the many colours of the rainbow around the South Island, haplotype G remains solely with Māui dolphins. For more about our updated Hector's work, go and see Debbie Steele's poster on Thursday. She really loves chatting to people endlessly. She would love you to visit. I can hear her laughing. Go visit her just to get back. By the late 1990s, we knew the Māui dolphin was clearly distinct from the Hector's dolphin, and it was deeply in trouble. Yet uncertainty remained, and this was exploited by some government officials, recreational and commercial fishing lobbies to delay protection, as so happens throughout the world. Our story is not a unique one, sadly. The Minister of Conservation proposed protection measures in 2001, which were vehemently opposed by the fishing lobby, who challenged the minister in the High Court of New Zealand. Legal action by the fishing industry saw scientists spending more time than they should in court defending what seemed like basic research. Whilst this was happening, 3 more dolphins died in set nets, and I remember vividly the day they came into our lab and were lined up on the floor. To continue delaying action, the authorities insisted that they were able to act for a number of unable to act for a number of reasons, mainly around there's uncertainty of how many dolphins there are, uncertainty of where they go. We knew there weren't many, we knew their range was retracting, and every year that passed without action, the situation got worse. The Māui dolphin is an example where uncertainty was an excuse for a lack of urgency, and the dolphins paid the price. So research was focused on determining how many dolphins there were, starting with aerial surveys in the early and mid-2000s. February 2000 was a milestone year because it remains the last known Māui dolphin bycatch in fishing gear. I'm not saying there hasn't been another one, but this is the last one that we have, and we know that bycatch killed it. 2003 was the first genetic abundance estimate using living and dead dolphin samples. There was a gap in knowledge about dolphin use of harbours, and from 2005 to 2008, acoustic devices were deployed. Will Raymond led the write-up of this work. This yielded extremely low levels of detections, with 97% of them coming from the dolphin's core habitat. Unfortunately, even though these acoustic tools are really valuable, the area is vast and it is such a dynamic space. The harbours are rapid flowing, the Tasman Sea, it brings westerlies in, and it's such a place that it's actually very difficult to do this acoustic work. And I know Will worked very, very hard to get the information out of this that he did. Finally, in 2008, after over 2 decades of acrimony and delay, the Marine Mammal Sanctuary, out to 12 nautical miles, was created using the Fisheries Act and Marine Mammals Protection Act to protect Māui dolphins, with set nets banned out to 7 nautical miles and trawl fishing banned between 2 and 4 nautical miles. So the protected area is 12 nautical miles offshore, yet the ban on fisheries is only a fraction of that. The irony was not lost on us, but you take your wins where you can get them. Better late than never, I guess. But the damage was done with almost 40 years of fisheries-caused mortality and time wasted arguing. International and local pressure had increased substantially. Newsflash, the Māui dolphin was in trouble, recognised by the IUCN as critically endangered, and it was on the International Whaling Commission agenda and in their sights. Research efforts increased with a focus on dedicated synoptic surveys during the broader summer months when the dolphins aggregate to breed, and the weather allows us to more safely undertake research in these dangerous and shallow west coast waters. I'll just go back so you can see. We often are out the back of a wave break and we talk to surfers about what they've seen, and we're even known to sit on our boat and have a conversation with people on shore. That's how close the dolphins are into shore. And with almost always a 1 to 2 metre swell, we could be as beached as some of the dolphins that some of us study. So it's tricky driving. Always grateful for our very good skippers. We've spent a lot of time on a small boat, driving up and down the coast looking for these dolphins. But almost all that we know is from the summer, and we really do need to focus on the winter months, but it is a challenge. Since 2010, we've conducted 3 two-year synoptic surveys to undertake a genetic mark recapture in 2010-11, 15-16, and 2020 and 21, with our 4th survey planned in 2025 and 2026. And no, you can't join us, the boat is full. You have to book a long way out, sorry. We photo ID the dolphins, but with only 15% of the individuals marked, and we're seriously pushing the definition of marked here. I work on bottlenose, and none of these would probably make the cut in a bottlenose catalogue, but these are some of our best dolphins. It's handy information, but at best it's mostly supplementary data rather than the primary method that we would use. These maps show where the dolphins were in 1 year of each of the 3 synoptic survey periods. You can see the range, but you can also see how clustered they were in an abnormally warm ocean in 2015, so the middle one there. And I'll talk a little bit about that. There was a big El Nino year, and New Zealand's been hammered by some really extreme warm ocean events, which are giving us some cause for concern. You can see here how that much their range has retracted. The protected area is in green, and the lines point to 2021, the extent of where the dolphins are. Their range is even more a small fraction of what it once was. All 70 samples collected between 1989 and 2007 were haplotype G, Māui dolphins, and then they got some friends. In 2010, we had a few surprise guests. 2 female Hector's dolphins, the I girl and the J girl, were found swimming in amongst the Māui dolphins. We never knew they were there. Both were from the West Coast South Island. We don't know where, but swimming in a straight line, it's about 400 kilometres. They were certainly a long way from outside their range. Hector's dolphins have a range of about 40 kilometres or so. So these 2 ladies really busted a move to come up to the North Island. We also have had 2 CA males who come from the east coast of the South Island, or maybe the top, but eastern side of the South Island. One of those males we've seen over the course of 2 years, and one of the females we've seen for several times over a period of 10 years. We've also genetically identified 4 beach-cast Hector's dolphins, most likely from the east coast of the South Island. 8 Hector's dolphins along the west coast of the North Island since 2010, and many of those are from the top of the south or from the east coast. In 2012, a Hector's or Māui was reported as bycatch in a commercial set net off the coast of Taranaki. Unfortunately, whilst fishermen must report captures in New Zealand, there's a clause in the law where they are not allowed to bring the dolphin back to land. You can imagine how excited we were to hear about that clause. So the dead dolphin was legally discarded at sea by the fisherman, and he reported it when he came back. There has recently been an amendment to that clause. So in the future, where they can land the dolphin, we'll never know what it was, whether it was a Māui or a Hector's, and sadly it was another missed opportunity. With Hector's and Māui mixed together, there's the potential for hybrid offspring, and we can easily detect those. To date, there are no hybrids, even though that one female, the J girl, has been sampled with Māui dolphins several times over the 10-year period. So let's see what 2025-26 brings us. Debbie is like a small child waiting for Christmas presents when she looks at the DNA, and she has a little sad face when we have no hybrid. There may be some other factors going on, and Alana Alexander down at Otago and her student Sebastian, who spoke yesterday, are looking into some of those things. We now know quite a lot about these dolphins, thankfully. Since 2001, we have genotyped 138 individual Māui dolphins, 122 when alive, 20 beach cast, and 4 were alive, and then we got them when they were dead. We have 4 males who we've known for around 20 years. We biopsy animals when they're older than 1 year of age. They grow very quickly as calves, so once they're 1 year of age, they're quite large. So these dolphins are certainly probably well in their 20s. Like the Hector's, Māui dolphins do not range too far. Their mean range is only 36 kilometres. But we did have one dolphin who ranged 86 kilometres, and forward, that one, this one. And he was sighted up here by Kaipara, and then down here, so about an 86 kilometre stretch. So that's the maximum distance that we have. In 2012, the bycatch incident of the Cephalorhynchus off Taranaki that I just described, and the new abundance estimate that we generated from the 2010-11 mark recapture, triggered the Ministers of Conservation and Fisheries to extend the fisheries exclusion zone out to 7 nautical miles, and you can see that here in the purple, on the dark colour and the purple. This was met with intense resistance by the fishing industry, as always, as set netting was a dominant gear type, but the change was made anyway. The 86-kilometre maximum range of the Māui dolphin up in the north there informed the new southern boundary. I think it's been the most unusual adoption of a piece of information that we've seen being written into law, but there we have it. You never know how your data are going to be used. The 3 synoptic surveys were used to generate the abundance estimates. We used genetic mark recapture, and in 2010-11 we had 55 dolphins. We had 63 dolphins in 2015-16. And in 2020-21 we had 54 dolphins. In 2015-16 and 2020 and 21, Justin Cook adjusted the estimate to account for mortality, which reduces the estimate slightly. But they are all there or thereabouts within each other. In 2016, sea-pod acoustic loggers were deployed offshore from the dolphin's core habitat, with successful detections, mostly near shore, of course, which was not surprising. But there were a few detections outside of the 7 nautical mile fisheries interaction zone, which is of great concern and is still something we need to address more fully. And today, this is our marine mammal sanctuary, the West Coast North Island Marine Mammal Sanctuary. So we have the out to 12 nautical miles zone, but then the devil is always in the detail. And when you look at it, you can see there's different amounts in harbours, out of harbours, set nets, trawl nets, the usual difficulty that we have. But this area that forms the whole of the sanctuary is an area of almost 21,000 square kilometres. The restrictions that we have are fine, but they are certainly not perfect. They do protect the Māui dolphin, but interestingly, they also protect those Hector's dolphins that are ranging north. And I think that that's an important thing. Even though they're not Māui, this movement by these dolphins is a very interesting and relatively recent phenomenon. With our new pro-fishing, anti-conservation government, we hope this protection stays. But to be honest, none of us are holding our breath. The decades-long curation of tissue samples and museum specimens has been critical to taking advantage of science innovations. Never underestimate the value of archives and archival information, and please always fight to maintain them. They're often the first things that get cut when budgets are tight. For example, we've used our old samples to work out how old is that dolphin. With the new ability to epigenetically age dolphins, we were able to take tooth samples that had been collected from back when Liz and Steve started their work, and then through necropsies since that time of Wendy and Porrig. And so we were able to use the traditional tooth aging method and combine it with these animals that we've known for so many years, that we've genetically identified. And then we could apply ages to these dolphins. And we can do it for living dolphins, dead dolphins, and look back in the historical records. And this is work that was led by Keith Hernandez and Debbie Steele. We can accurately age the dolphins to around about 1.5 years. The median age of the dolphins in 2020-21 was about 1 year younger than the dolphins in 2015-16. This may be a consequence of the establishment of the Marine Mammal Sanctuary in 2008, which largely removed the primary threat, the risk of bycatch. Individuals born after 2008 and their parents would have a greater chance of survival, as with the offspring. Or it may be an artifact of fluctuations often seen in very small populations of any animal. Future work may resolve some of these questions. But it certainly has been a really important and fundamental leap forward in our ability to think about the Māui dolphin. Māui dolphins prefer turbid waters, targeting small prey throughout the water column. We very rarely see what they're feeding on. So we use stable isotopes to determine their diet. This revealed that before implementation of the sanctuary in 2008, the dolphins worked harder to find their prey, as evidenced by the larger ellipse area compared to that shown after 2008. With the removal of fishing pressure, maybe there's some ecosystem restoration, but unfortunately there's no monitoring of the ecosystem in this area or looking at what happens in the system when fisheries is removed. In 2016, the dolphins had to work harder to find prey. Remember, that was the year where they were all clustered together very tightly. Scott and I sat on the boat one day and we could see the furthest north and the furthest south that the dolphins were, and we counted 32 dolphins within our sight. And Scott and I were on the boat, and we were very, very, very concerned that a meteorite struck right then. There would be a big loss, not only of 2 researchers who knew all the old history, where the skeletons were, but also, more importantly, where half the population of Māui dolphins were. They were clumped together really tightly, and they were working very hard to find prey, similar to pre-2008. We also saw, though, a population-level shift in diet, and it was most likely due to the arrival of a novel prey coming down on the super warm water. This diet flexibility was hugely reassuring, and with ocean warming increasingly an issue, we're really kind of I guess, in some ways happy for this diet plasticity, and hopefully the dolphins will make it if their preferred prey are no longer around. We're already seeing shifts in dolphins' primary prey moving out of their core habitat, so having a flexible diet is pretty important. We now look to the next phase of a revision of the threat management plan that's underway, and it's being led by Jim Roberts, and we're focused firmly on the future. Following on from the aerial surveys of the 2000s with people in a plane looking out the window, we've been working on aerial surveys and AI. We've been testing a large drone, so this is Tane, Willie and I, who started a charitable trust, Māui 63. New Zealand no longer has a good fit-for-purpose aerial survey aircraft, and there are safety concerns about putting people on small aircraft. So having remote, either a drone or a plane with camera rigs under it is sort of the way of the future. We've trialled this large drone. We're able to fly it beyond visual line of sight, so we can fly some distance away. There are challenges with this technology, but the development of AI has worked well because the Māui dolphins are very distinctive. The convoluted neural network model has around 90% accuracy and gets better every time we add more data, of course. So we can detect these 1.4 metre dolphins in the very murky ocean. Where's Māui? On this big screen, you should be able to see it. The people who never leave labs are excused. There it is. Unfortunately, these days, we often see single dolphins. It breaks my heart, in particular, when I see a mother alone with her calf, and it happens more than is acceptable. They should not be alone. But with so few dolphins, their groups are typically only a few animals these days. We have broken their society, the very fabric of what it is to be a social delphinid. It should come as no surprise that Māui dolphins are very closely related. There's not a lot of choice on the West Coast. People I know who live on the West Coast would agree with that, but for different reasons. But they really are very, very, very related. And we're moving really to genomics to tell them apart. We're using dd-RAD sequencing to understand their pedigrees, who's contributing to the gene pool, and there's some really interesting patterns emerging, and I'm not going to steal the thunder from Keith Hernandez, and he's going to be talking about this on Thursday, so go along. Unlike Debbie, Keith really loves talking about his work, so go see Keith's talk. It'll be fascinating, and it's going to make a real difference to how we understand these dolphins. Wendy Rowe has continued the work started by Parag Dignan, determining the cause of death of 24 Māui dolphins collected since 1997. This has shown that disease is the most common cause of death. Shark predation killed 3 dolphins. Bycatch killed 3, as I mentioned, the last one in February 2002. And we have challenges with advanced decomposition or incomplete records, and there's one awaiting permission from mana whenua, from local Māori, to be handed over for Wendy to work on. As you heard yesterday from Jodie and Charles, they are their relatives, they are family members, they are their ancestors. And so for Māori to hand these dolphins over for us, even just taking a small sample or to do a necropsy, we are hugely grateful for their trust in the process, but they know how important it is that the Māui dolphin come back. So what about the fishing risk? Well, it's not zero, it's never zero. Even though the set net and trawl fishing is banned between different distances offshore, occasionally there are visual or acoustic detections of dolphins out to beyond these distances, as I mentioned. Unfortunately, there are no samples of these animals to confirm species identity, and acoustically, Māui and hectors sound the same, much like they look the same. But we do know that the bycatch risk is not zero for as long as dolphins are ranging beyond those protected zones. After many years of delays, as often happens, cameras are finally on fishing vessels. The irony is not lost, I think, on any of us who's had the fight with the fishing industry to have cameras on vessels, because when you walk down any street in almost any town or city anywhere in the world now, you are captured constantly on cameras. It's part of the fabric of life. Yet it delays, I guess, meaningful action to protect vulnerable animals. Thanks, buddy. There have been 2 recent court cases of boats fishing in the sanctuary. There was a trawler seized and a $14,000 fine, and there's a commercial set net fishing situation that's in court. As I explained earlier, until in an interesting turn in 2020, Sea Shepherd USA took the New Zealand government to court to ban fisheries imports into the USA based on poor protection for the Māui dolphins. Unfortunately, this case was based on misleading information. As I explained earlier, until 2023, when we got a single sample, we had no genetic evidence of whether they were Māui or hectors dolphins along the east coast of the North Island. The Sea Shepherd legal case claimed that all North Island sightings as Māui dolphins. If successful, it would have had major consequences for the New Zealand economy. It's been in and out of court a few times, this case. These kinds of actions are not helpful, and discussions, even with uncertainty, should always be honest. The question marks largely remain. Cats kill dolphins is always an interesting conversation starter in New Zealand, but it's a conversation we can't shy away from. 3 Māui dolphins have died from toxoplasmosis, the active disease as a result of infection by the protozoan Toxoplasma gondii. Toxoplasma can only complete its life cycle inside a cat, where the oocysts are expelled in cat feces. Cats are an introduced species to New Zealand and are an invasive mammal. We have high levels of pet cat ownership, but also many feral and stray cats. There's a lot of discussion about this anthropogenic threat to Māui dolphins and what action is needed. A toxoplasmosis action plan has been developed with targeted funding to understand the source of infection, feral cats or pet cats or both. Also determining how many oocysts are making it into the ocean and what strategies could be used to control movement. Finally, this is a social license issue, and social science is really important if we are to bring the public along with us. It says 3. What? It says 3, he says 1. Almost done. So this is Scott and crew on a small boat. This is 25% of the Māui dolphin population in this image. It's a confronting image. I show it, I love showing it, because this is not okay. Of course, there are many gaps in knowledge. This question list should go on, but we must remain future-focused. The dolphins are hanging in there. Since 2010, the estimates are similar. We know so much more than we did to inform future action and models. With new research tools in our toolbox, communities are engaged in addressing the issue with toxoplasmosis, and future action and addressing new challenges, I think, is going to need to be an all-of-nation action. We've wasted a lot of time because of inaction due to uncertainty, and it has to stop. It'll take an epic amount of effort and a sprinkling of luck to save the Māui dolphin, but you never know, they might make it. I'm going to finish with some footage of them at their most Māui-ness, happy in the murky water. As I said, it's not Shark Bay. It's a very hard place to study, these dolphins, but it is such a magnificent place. Thank you so much.
Thanks very much, Rocky. I think we're going to start on questions while it plays, if that's cool.
Yep.
Yeah. So we've got about 4 minutes for questions if people want to get the details from Rochelle. You know where the mics are.
Oh no, someone's moving.
Hi, thank you for a wonderful moving talk. Obviously, you're seeing calves coming into the population. Do you think that this is a normal expected amount of recruitment, or do you think there may be problems in that area?
Yeah, we did some work on progesterone. Emma Carroll was involved in this work, and we found that they were pregnant at a rate you would expect, so one calf about every 3-ish years, so that was good. The calving rate is it fluctuates, so some years we see one, we'll often only see one, and then other years we'll get 3 or 4, which is reassuring. So yeah, I think when the population's this tiny, there's a lot of stochasticity that is hard to account for, but hopefully modelers will do some magic with it. But yeah, they do have babies, so that's good. I mean, the vaquita even has a baby, and there's, so no matter how bad it is, sex still happens, eh?
Amen to that.
Over on the other side here. You can wait, Michael.
Hi, Rochelle.
Hi.
Thanks for the confronting but excellent talk. I was just wondering if you wanted to comment on what we as a society of interested scientists and concerned scientists can do to help, if anything.
Yeah, it's a really good question. I'll be really frank, I think people need to stop picking on us. We get, New Zealand is pretty poor, relatively speaking. These animals are deeply in trouble. And one of the big challenges I think that's always been is that it doesn't matter how hard we argue, kick, scratch, and fight, we're not going to get more money. It's just a reality, and I think it's the reality of many in this room. So we work very hard on very little money, and the government will only put so much into it. So one of the things that's been quite difficult is getting these international, large, well-funded nations telling us that what we should do. It's like, well, you can write us a check, I can send you our bank account, and you can put the money in there and we'll do it. But we genuinely are working our hardest to do it. Pressure on the government, yes, that works. I think that's probably the most important thing. But having a crack at the poor science when we are all doing our best is something that doesn't help. So I know you weren't all coming for that. You're all wanting like, ooh, write a letter. No. Genuinely, I think we need to come together as a society, and if there's anything for all of the countries and research things where we have really wicked and urgent problems is maybe to offer support. What do you need? How can we help you? Even for countries like New Zealand, how can we be your ally, not just criticizing you? I would argue as a society, For people who work, so like I work on humpback whales and bottlenose, like the white rats of the cetacean world, big and small, right? And so there's so much work, and it's awesome, and it's like Richard was saying, there's so many things we can learn from these long-term studies, but sometimes step away from your comfort zone of the thing you know well, and maybe think laterally, and get to us, say, hey, I've been thinking about what I have, and maybe have you thought of this, and can I help? I know that sounds probably an odd answer to your question, but we're all for ideas. We are open. I will hand our data, Scott, Debbie, all of our data to anyone if you can help us find an answer to making this, the Māui dolphins survive. So yeah, just get in touch. Don't lobby grenades at us. We're really trying our best. Thank you for asking.
Over to you, Michael.
Great talk, Rocky. Thank you very much. You might remember I'm very closely connected to Hector's as well. I was probably the first person in the world who actually darted Hector's dolphins. My question is also, especially sort of in line that sometimes I feel that a committee for taxonomy in the society is rather conservative. To what extent did it help that Maui was elevated to a species level? To what extent did that help the conservation of the species?
Yeah, it's a great question. So there's splitters and joiners, Michael? We have this conversation often. So Alan Baker, who had worked on the Māui dolphins for many years, there are two Bakers. There's the Scott Baker and the Alan Baker. So Alan was very deliberate in getting them named Māui dolphins because it was a marketing exercise. He's like, no one's listening because they're North and South Island hectares. It sounds like they're the same. There were clear differences. There's some physical differences and, of course, the genetic differences, which we know are, substantial. It was the last glacial maxima that, separated them. But it was a marketing exercise to get the world to pay attention, and it did work. But there are genuine reasons for it, so please don't go out and it's not the latest social media campaign to try and decide that your species is different. But yeah, it had a double purpose, and by being called the Māui dolphin, that really helped. With Māori being engaged in it as well. But most hilariously, the Hawaiians were like, why did you name it after a Hawaiian island? So we had a little discussion about global gods and, the big Pacific Ocean. Yeah, but yeah, it was very important. It's legitimately good, but it's had a very, very big impact. It's a good question.
Okay, we'll have time for two more quick questions if possible.
And two quick answers, Rochelle. Yes.
Thanks so much for a wonderful talk. My question is kind of related to that last one about species status. I wanted to ask about the hybridization you were talking about. I know you mentioned it would be really exciting from kind of a novelty perspective, but I naively wonder if it would potentially kind of lose the pure Maui dolphin genotype if enough hybridization would take place. Is that something that you are worried about or think might happen? And what's your perspective on that from like a conservation of biodiversity perspective?
Yeah, it's a really good question. We've had this discussion a lot. The fishing industry really wants this kind of genetic rescue by hectares and hectares to come up because then they're like, oh, well, we can just go fishing again. No, not okay. I think the idea of hybridization and these splitting, we split them, right? These are our artifacts. These are not, these are our artificial constructs about how we decide taxonomic status. They are very useful and valuable. I don't think it matters. If they can interbreed, then cool. It's not like there's going to be 500 of them tomorrow. They're still in trouble. I'm an ecologist, and so for me, it's about having a functional population of a top-order predator along that coast again, because that's a big loss to that ecosystem. But it is an interesting thing, and there's a lot of really cool work going on. Like I said, Alana, Alexander, and Sebastian, who's here, around maybe they can't. Maybe there's some region in the genome where they can't. So Emma Carroll and Debbie and Scott are all working on that conversation. So they may actually not be able to interbreed, which will be pretty interesting for the genomicists of you out there. Thank you.
Hi, thanks for your really moving talk.
Thank you.
You mentioned some specialised legislation, the Marine Mammal Protection Act, and also some court cases. What role has the law played in your conservation efforts, and do you think it's been effective?
What role has the law played in our efforts? Well, there wasn't any, and Steve and Liz have kicked and screamed and yelled their entire career, and they're still doing it, and they're retired. I'm very proud of them. But genuinely, there was nothing in place, and what was in place was being ignored. Fisheries is more important than anything else. Money's more important than anything else. So it has been a real challenge. I think the most important thing is that the research has continued on, and we're doing the best research we can, and where we can, we've pushed it out to try and inform decision making. And I think it's really important to protect the science as scientists, when you're working as an advocate, which you have to be when you're in this much trouble. So, it's very important, I think, for the science community to always protect what we have, to make sure that decisions being made are research informed and don't have unintended consequences from that. But like I said, our current government is making us very nervous, and they could just undo all of this. So we go back to square one, and that's why capturing hearts and minds is so critical. If politicians are only in because people vote them in, and politicians will pretty much do what the people want. So saving the Māui dolphin is a team sport, not just in New Zealand. I think it's a global team sport, so we have to keep going. Thank you.
Thank you.
Please join me in thanking Rochelle for a great comment. Cheers.
Just a very quick little bit of housekeeping. Timing can be a little off at these things. There's a lot of pressure on 5 concurrent sessions. Importantly, chairs are supposed to stick to the schedule. Not mentioning any names. Nick Gales. Nick. As crowd members, you can speak up too. We don't want to go ahead of the schedule. It's okay to go a minute or two behind. We don't want to go ahead of it. This should happen in long talks. This should happen in long and short talks. And then this. Don't get hippoed. It's now time for another break. Enjoy some coffee and a snack, and I believe it's 11:30 back here for the Wood and Norris Awards. Thanks so much for attending. We'll see you soon.